Metzger. I'm the curator of the Massey Herbarium at Virginia Tech. So I work here taking care of the plant collection in the herbarium. I work with a lot of students on different research projects. We'll be seeing some examples of those in a little bit. And I also do some teaching, teaching plant taxonomy to about 70 students right now. So do a little bit of everything along with some outreach here. But I'll do a quick presentation about what the herbarium is. I know some of you have been here before, but some of you are probably new to it. And then I'll give a quick little demo of our online specimen database to show you how that works. And then we have video clips from the students. If you give me a second here, I will share my screen. Okay, so the herbarium, we're over here in Daring Hall. So right smack dab in the middle of the new construction zone here on campus. But an herbarium, if you're not familiar with the word, it's basically just a fancy term for a plant museum. So we have dried, preserved plant material here. So we have, a greenhouse on campus that has a living collection, but this is all preserved material. So basically, it's the botany wing of a natural history museum, if that's easier to think about it that way. Worldwide, there's about 3,000 herbaria. So that map just kind of shows where they're concentrated. There's a lot of them in Europe, in the US, and in China as well. But they're found just about everywhere. And herbaria have been around for over 400 years. The first ones were started in Italy in the 1500s. And since then, all of these herbaria around the world have collected over 350 million specimens. So this map is showing how many specimens we have from different parts of the world. A big circle means we have a lot, a little circle means we have a few. But basically anywhere there's dry land on Earth, even down at the bottom here in Antarctica, we have herbarium specimens from that area, so we can document the flora that's found there. So that 400 years is kind of an important figure for what we do here. Some of those specimens from the 1500s are actually still around, not in our collection, but over in some of those herbaria in Europe. So this photo here, this is a tulip that was collected in the 1500s. The photo was taken about 10 or 15 years ago. So you can see that it's still around, and for being 400 years old, it's in pretty good shape. So basically, if we take care of these things, they'll last forever. So our goal here is to preserve this material indefinitely. We want Virginia Tech students or researchers to be able to come in the year 2500 and be able to use our specimens here. So we use a lot of what are called archival practices. For us, a lot of that comes down to the materials that we use. We use paper and glue for preparing the specimens that's acid-free. So if you think of an old newspaper that's brittle, that's not archival. That has a high acid content. So the paper and glue and things that we use is much more stable. We try to control the environment too. So things like light exposure is bad for the specimens. It fades them really quickly. The humidity is important. We have to be on the lookout for insect damage. So we keep an eye on all of those things in the collection. For example, some of these are pretty low-tech solutions. I have bug traps around the room that I check every month to see if insects have started to move in and chow down on any of the specimens. Some of these are more high-tech. We have a digital archival standard now. It's called the Darwin Core, and it specifies how we enter data into our database. So a good example is thinking about how you enter a date. So if we were going to database a specimen here and it was collected on July 9th, we would enter that as 7/9. Then a researcher in England would see that and think that we meant September 7th. So, so the Darwin Core format specifies even how we enter a date, so that there's no confusion over it, and that different collections can talk to each other and understand what the information is. Oh, here's an example of some of those physical things like the bug traps, and this little white box here is a little data logger that we have stuck on one of the cabinets, and every, I think, hour or so, it records the relative humidity in the room, the light exposure, and the temperature. Our conditions are usually pretty good in the herbarium. We don't have to fiddle with things too much, but if something happened and the building conditions changed, we would be able to show our campus facility services, the humidity has been spiking in the room, and we can show you that it happened a month ago and it's gone up this much, and we would be able to get that resolved then. We also keep the specimens in these big steel cabinets, and these cabinets protect the specimens from a lot of things. They keep out light when they're closed, but then they also have seals that'll keep out insects. And if one of the water pipes in the ceiling breaks, it gives us a little while until the water starts to leak into the cabinet. So they really help us with protecting the material. So our herbarium here, it's called the Massey Herbarium, and we have this little abbreviation that we use a lot, VPI. Comes from Virginia Polytechnic Institute. There's a little basically phone book called Index Herbariorum for herbaria around the world. Every herbarium registers in it, and they do some kind of letter code like this, like 1 to 5 letters. So ours is VPI. So when somebody mentions our specimens in a paper or something like that, they do it as VPI. But our herbarium was founded by A.B. Massey. That's his picture there. He was a microbiologist here, and he became interested in botany and shifted his research focus to that. He was involved with a lot of the early efforts to map the Flora of Virginia. So a lot of his time was spent trying to map out distributions at the county level. So in the modern times, that's basically become the Virginia Digital Atlas website that a lot of you have probably used, with the county maps that have a dot for whether or not the species is found there. Our collection, this slide is actually a little outdated. I think we're up to 119,000 specimens. Most of those are vascular plants, so things like ferns or pine trees or anything with a flower or fruit. We're up to about 3,000 mosses now, and we have over 5,000 fungi. We had a mycologist here a couple decades ago, Orson Miller, who wrote some popular field guides and things, and him and his students collected a lot of mushrooms that we still have here. Our lichen collection is the smallest one. We're still at 800 or 900 lichens, so we're trying to get a little more activity in that collection, but it's the smallest one right now. So 119,000 specimens, that makes us the largest herbarium in Virginia. In the state, we have 26 herbaria with a little over 500,000 specimens. So about 1/5 of the total holdings here are from Virginia Tech. The other big ones in the state are places like Lynchburg, Longwood, William and Mary, and George Mason. So we are the largest in the state, but that 119,000, we're still kind of a mid-sized collection. If you go just over the line into DC and went to the Smithsonian, they have 4 million specimens. So they have kind of a global focus. With our collection size, we're much more focused on Virginia and the surrounding area. Some of our rarities or unusual specimens include this sedge that was collected in New Jersey. It's Cyperus diandrus, and it's actually our oldest specimen. This one was collected in 1829. So you can see a little bit of the original label down here at the bottom. In 1829, Hackensack, New Jersey, in Torrey's handwriting there for the original collection. This is the specimen today, so it's still around and still quite usable. The other neat thing is that this shows how we identify our specimens and how we keep them up to date. So there's this little inset photo here that zooms in on the bottom right of the specimen. And you see the original label down here from 1829. The collector put Cyperus fulvescens, but he wasn't totally sure, so he put a question mark after it. And then in 1972, Leonard Utall, who used to work here, wrote Cyperus fulvescens on it as well, so he was agreeing with Torrey. And then 10 years later, his successor, Tom Wiebolt, looked at it and said, this is actually Cyperus diandrus. So he put the new identification on it. And then in 2000, a sedge expert at Valdosta State looked at it and said, yeah, this is Cyperus diandrus. So these are called annotation labels, and they're how we keep our material up to date. So this way, if we have an expert visit the collection, or if we send them specimens to look at, they can identify them for us, and we can keep our material up to date. This is Micranthemum micranthomoides. Doesn't really have a common name, but this specimen was collected in the fall of 1941, and it's actually, the species as a whole has never been seen again. It was collected in the Chickahominy River. It had this weird habitat. It would grow in the intertidal zone of freshwater rivers, so pretty narrow habitat requirement. And it was found from Virginia up through Maine, and was never really thought to be a rare plant or anything. This collection was made just as kind of part of a normal collecting effort. And then years later, people realized no one had seen the plant since then. And there's been efforts to look for it, and no one's been able to rediscover it. So at this point, it's presumed to be extinct. The thought is that there was probably some disease or insect pest that suddenly came through and wiped out the species in a short amount of time. I will say, if you look online, you'll find people selling this for the aquarium trade, but anytime it's been, somebody's checked one of those, it's been a different species of Micranthemum. They're very small plants and pretty hard to tell apart. A little closer to home, this is Iliamna cori, Peters Mountain mallow. So this grows over on Peters Mountain in Giles County. That's the only place in the world where it grows in the wild. So this is a specimen from a greenhouse plant. We were involved in some of the conservation efforts for this species. So some of the staff here back in the early 2000s was growing this in the greenhouse and studying it and trying to help with the reintroduction efforts. So it's still restricted to that one mountaintop in Giles County, but the population, I think, is up to a couple hundred plants in the wild now, from the 5 or 10 back when this project started. So I mentioned that we focus on Virginia. Almost all of our materials from the USA. We have very little from overseas, and around 2/3 of it is from Virginia, and about another 1/4 of it is from states right around Virginia. So we're really focused on our local flora. So if you want to understand or learn our local flora, we're a great resource. If you want to research like the plants of Fiji or something, you'd be better off going to the Smithsonian for that. If you look across Virginia, a lot of our collecting is pretty local. So this is a heat map. The hotter or more red a county is, the more specimens we have from it. So you can see Montgomery County here is the darkest red one. And things get a little cooler as we go towards the coast. So we do a lot of collecting locally, and if we can make it there and back in a day, we're much more likely to have collected specimens from that county. This is a cartogram, just a different way of looking at it. This changes the size of the county to the number of specimens. Sometimes you see these on election night for like how many votes a district has or something. So now Montgomery County is this huge thing down here. So you can see that the mountains are looking pretty fat. So we collect the mountains pretty heavily. They're local and they have a lot of neat microhabitats. We collect the coastal plain about what you would expect on a land area basis. And then we lag behind in collecting the Piedmont. So we don't seem to have as many specimens from the Piedmont as you would expect from the land area it covers. Specimens here get used for a lot of different things. Just thought I'd mention a lot of the illustrations in the Flora of Virginia actually come from our specimens. Most of those were illustrated by Laura Call-Gastinger, who lives in Charlottesville, but she's a Virginia Tech alum. So for example, the black walnut and butternut illustrations were based on our specimens as models. And I'll just give a quick plug. Probably most of you are familiar with it already, but the Flora of Virginia now has an app version of the book, includes an interactive key that a lot of people have found pretty handy. We do a lot of outreach too. So a lot of that is geared towards younger audiences in schools or at science festivals. But we work with adult groups too. Last night I was doing a training session for the new Master Naturalist class. So we do a lot of outreach efforts here as well. Research is done a lot by students and our staff as well. I'm the only official staff member at this point, but I still count. And then we work with outside labs too. And we actually just published a student paper right at the end of 2021 that was summing up a student project out in Stadium Woods. We had a student, Alan Milby, who was collecting mosses and liverworts in Stadium Woods and also Center Woods on the edge of campus. So it was nice to be able to publish something and show how Stadium Woods is important for student research here at Tech. So as far as where our material comes from, we collect some of it ourselves. We get material from other labs at Virginia Tech too. For example, the Poison Ivy lab had some voucher specimens documenting some of their research sites. So they brought us a big box of poison ivy one day. So we added those to the collection. We get some from government agencies, a lot from the Natural Heritage Program in Virginia when they're doing their rare plant surveys. And we get some from private collectors as well. And then otherwise, we do a lot of exchanges with other collections. So we might send a box of material to another herbarium, and then later they'll send us some extra samples of their local flora. And those loans, we do those between different institutions. So for example, this is showing a loan from the Academy of Natural Sciences for one of my students to work on. And these are kind of temporary things. They send you the specimens, you have them for a year or two, and then you send them back. You might use them for trying to identify them, for mapping a species range. You might use them for like a DNA research project. You could use them for some of those botanical illustrations, all sorts of things. And the presentation's froze on me here. Give me a second. I'll just mention some of the identification apps that we found have worked pretty well for people. You're probably familiar with a lot of these already, but iNaturalist and Seek are nice free options for your phone. And that Flora, Virginia app that I mentioned earlier, it does cost about $20, but it has that awesome graphic key that lets you make choices based on where you're at. So it can use the location of your phone, and then you answer questions like, I'm in a wetland in full sun, and the plant I see has opposite leaves and red flowers. And it'll spit out a list of all the species known from that county that occur in that kind of habitat and have those traits. So it can be a really handy ID tool. These are some of the other sources that are pretty helpful, like the Digital Atlas of Virginia, and some Facebook groups are also pretty helpful, like the Virginia Native Science group, or the discussion group for Alan Weekly's Flora. A lot of people post pictures there and get IDs. The last one listed is Cernak. I'll just show you that real quick. That's our collection database for our vascular plant specimens. So we use all these museum databases that are set up with other universities for our collections to put things online. And Cernak is the one that we use for our vascular plants. So Cernak stands for Southeast Regional Network of Expertise and Collections. And it's a project with 233 herbaria across the southeastern US. So altogether, all of us have databased over 3 million specimens. And usually for a specimen, we make a record for it and then also try to upload a high resolution picture. So it can be pretty powerful. There's a lot of information there, but it is pretty tricky to use. The user interface isn't quite as friendly as like iNaturalist or something like that. So I can show you an example on the browser in a second here, but in general, if you want to try using Cernak, and the link is there, I can put it in the chat in a minute. You want to try to use very few search terms. So put in the species that you're after, and then if you want to narrow it down, you could put in the state or maybe the state and county, but I wouldn't go any further than that. If you try to put in like the collector who made the specimen, or oh, that's supposed to be date, not data, or the collection date or GPS coordinates, those usually aren't entered for most of the collections. So it's more, you get better results if you use fewer of those search fields. You can actually limit it by herbarium if you want. Like you could just search our collection, or you could search all 233, depending on what you're after. And when names change, sometimes Cernak will search both of those names for you, the old one and the new one, and sometimes it doesn't. So that's another thing to kind of keep an eye on. But I will switch over to the search screen real quick. So this is the Cernak homepage, and the specimen search up here on this tab, you want to go to search collections, and then it'll bring up a list of all 233. So as a default, you can just come over here and hit search if you want to search all of those. For this example, you hit this, it unselects everything. And then we'll go down and we'll just choose the Massey herbarium. So we'll choose one of my favorite plants, walking fern. When you start to type in a name, it gives you these suggestions, so that way you don't misspell it. So that's a pretty handy tool. And then we'll just see what we have total. So that's just searching on the name Splenium rhizophyllum for walking fern, and just searching the Massey herbarium. So that brings up 91 records. Walking fern used to go by the name Camptosaurus rhizophilus. And you can see here, they've got 2 synonyms entered, but they don't just have Camptosaurus rhizophilus. So if you search the old name, if you entered this full thing here with the form, it would pull up the records. If you just entered it as Camptosaurus rhizophilus, it wouldn't pull them up. So it gets a little tricky when the names change. But each of these rows here is a an individual specimen. So this one's pretty typical, so I'll hit the details on it. I guess I have to switch to that window for the screen share. So this is pretty normal. Because we have so many specimens, we're just creating records that only have a little bit of the label data on them right now. So it has the species name, it has a tracking number, and it has a little bit of the location. So you notice the date's not there, the collector's not there. None of that's information is entered. And then you can see these images here. And if you just want to look at it on the browser, the medium one's usually fine. If you really want to zoom in, you can go to that full resolution file. It's just a lot bigger. It takes longer to load. But you can see the label for it. If there's more information on there, like date or location, you can get it that way. And then if you want to see how to identify the plant or look for some particular character on the plant, if you can see it with the naked eye, you can usually see it with one of these photos. It is kind of tricky, so if you ever want help with it, just let me know, and I could work on it with you. So that's it for the little herbarium intro. Can move over to the student talks now, unless you have any questions. But originally, the plan was to have the students come and present live to us, but unfortunately, I didn't realize that this was going to be Virginia Tech's spring break week. Last year, they didn't do a spring break week, so I just forgot about it. So the students recorded their presentations before they left town, so I can show those, the videos that they made. I figured I'd probably start with the one on the cactus of Virginia, because everybody thinks it's pretty cool that Virginia has cacti, so it's a nice one to start with. So this is from Elizabeth Sicking. She's a bio major who will graduate later this year. I'll hit play and let her tell you about it.
Hi, my name is Elizabeth Sicking. I'm a junior at Virginia Tech and a biology major. And today I'm going to tell you a little bit about Opuntia, or the prickly pear cactus, and its presence in Virginia, specifically how to find and ID and differentiate the Opuntia hemophilia complex. So when you think of cacti, you will probably think of the really tall saguaro cactuses common in southwestern deserts and other really prickly, really spined cacti that thrive in environments with very high temperatures and low precipitation. But there's actually a cactus species in 46 of the 48 lower continental United States, and Virginia is one of them. In Virginia, it's the eastern prickly pear. It's a cold hardy cactus, so it can survive in pretty freezing low temperatures, as well as varying levels of rainfall. And the eastern prickly pear, which is also known as Opuntia hemophilia, is not one species, but a complex of 3 species that were grouped together because of their location. So those 3 species are Opuntia cespitosa, Opuntia mesacantha, and Opuntia hemophilia. And as of right now, we're not sure exactly how these 3 species came to exist in the same place, but one guess is that hemophilia is a hybrid of cespitosa and mesacantha. Before I get started characterizing each of the species, I wanted to mention the difference between spines and glochids, because they're 2 characters that I'll mention a lot. So as you can see in this picture, it's really blown up. But the glochids are the much smaller prickles found at the base of the spine and in the areole, which are the holes that are found across the pad of the cactus. And the spines are the much larger modified stem that you can see pretty easily with the naked eye. Glochids are small enough that if you want to get a good look at them, you're going to want some magnification, but just to get an idea of what they are. So we'll jump right in. Hemophilia is our first species we're going to talk about. This is the only species of the 3 that doesn't have any spines. That's its main identifiable characteristic. It has fully yellow flowers, as you can see in the picture, and its glochid color, which is another characteristic I use for identifying, is pretty variable. So it can range from anywhere to red, light to dark brown, golden, or even a mix of all of these colors. And it's most commonly found in the Piedmont region, so kind of central Virginia. Next up is Mesacantha. Like Hemophilia, it also has fully yellow flowers, but this species has spines, and its glaucids are consistently a light brown color. When you're looking, if you see a cactus in bloom and you're trying to determine what it is, if it has yellow flowers, you then want to look at whether it has spines or not. If it does have spines, it'll be Mesacantha, and that's the main way to differentiate it from Hemophilia. And it is most commonly found in the coastal plain region and kind of the eastern side of the state. The last species is Cespitosa. It also has spines like Mesacantha, but its flowers differ in that they are red tinged at the base of each petal. And the glaucids are also very distinct and have a dark crimson red color. And it can vary from a darker red to a brighter red and occasionally a dark reddish brown. And it's most commonly found on the western side of the state, kind of west of the Appalachians and in the Appalachians. To go and compare these characteristics a little further, we'll start off with spines versus no spines. You can see here Cespitosa and Mesacantha with the clear white kind of grayish spines, and Mesacantha that's lacking the spine, or Hemophilia that's lacking the spines. You can also notice, I'll talk more about glaucid color later, but you can see in this picture the reddish glaucids found on Cespitosa and the lighter brown glaucids on Mesacantha. Flower color, we see Cespitosa again with the red tinged base, and Mesacantha and Hemophilia are pretty much indistinguishable unless just based on flower color. So you really need to look for the presence of spines if you want to tell the two apart. Glaucid color, we have Mesacantha on the left, Hemophilia in the middle, and Cespitosa on the right. Mesacantha is that light brown I described earlier. This Hemophilia is kind of an orangey browny color, and this Cespitosa is actually not the best example of the color, as it aged a little darker than it is in the wild, but you can see that they all, the two that have spines, Mesacantha and Cespitosa, do vary a lot in color. And although it might appear that they differ in size, it's not actually a consistent difference in size. This just happened to be a nice little step kind of setup. So what I'm doing for my research right now is basically just IDing a lot of different herbarium specimens. You can see that Mesacantha and Cespitosa are very similar just in their specimen form, especially because you're often lacking a flower, and flowers also aren't really a reliable characteristic to use when looking at herbarium specimens, just because when dried, they all age to be a pretty brown color at the base of the flower. You can kind of tell that the tips are yellow, but at least if you were comparing like a Mesacantha flower versus a Cespitosa, you wouldn't be able to see that red based petal in Cespitosa because it all would age to the same brown. So it's really important to pull out the glaucids and look at the glaucid color, which is what I do using a pair of tweezers. Pull out glaucids, look at them underneath a microscope and good lighting, so I'm able to make sure I can tell what the color is. But those are the main characteristics I use for identifying the spines and the glaucid color. So the whole intent of my research is to establish the distribution of each of these three species within Virginia. To start off with, we have Cespitosa, which unfortunately we haven't seen a lot of specimens of, and the one we have seen was found in a western county, which follows its known distribution. Hopefully we'll get more data on Cespitosa and be able to map it out a little further, but as for now, we know it definitely occurs in the west, and hopefully we'll see a further occurrence. Mesacantha, as I stated earlier, is mostly on the eastern side and especially the coastal regions. We have some more data on this, which is nice because we're able to see its distribution a little more clearly, but it is mostly concentrated on the eastern side. Hemophilia, unlike the other two, is fairly evenly distributed over the state. This might be due to the fact that it's a hybrid between the two. So where they meet is where it occurs as well as where they both occur. So we see it on the west, in the central areas, as well as the coastal regions. All this to say, we're still hoping to get more data, and one of the ways we're doing that is through an iNaturalist project, which is a collection of all of the relevant Opuntia observations in Virginia. So what we do is we go through and add correct IDs where we can, and also filter out observations that shouldn't be there. But this is one way that you can get involved and help with our research, and by giving us more data to use. So if you see a cactus in the wild and you think you can ID it and add it to the project, that's always great. But also if you wanted to go in to the project and try to add accurate IDs based on what you learned, that would also be really helpful, as there's quite a few to go through, and that's something we're still working on. But yeah, I'd like to finish up by thanking Dr. Metzger and all of my lab mates, as well as Lucas Major, who's the researcher in Florida, who kind of started the differentiation of all of the Eastern Opuntia species. And then here are all of the herbaria I got loans from. But yeah, thank you for listening.
So that was Elizabeth's project. She'll be getting a bunch more herbarium specimens soon, and then hopefully over the summer we'll be able to do some collecting to flesh out those maps a little bit more. The next student is Olivia Madigan. She's a wildlife conservation major here at Virginia Tech in her senior year, and she's looking at climate change impacts on some of our spring ephemerals. So let me start her video. And
hi, my name is Olivia Madigan. I am a senior at Virginia Tech. Wildlife conservation is my major, and I work with Dr. Jordan Metzger in the herbarium. And my research is that I look at the impacts of climate change on Southern Appalachian wildflowers. So the two flowers that I looked at were Sanguinary canadensis, which is commonly known as bloodroot, and Trillium erectum, which is also commonly known as red trillium. Both of these flowers are commonly harvested and do have medicinal properties. And we just wanted to see what we could come out of it. And you may be wondering why bloodroot is called bloodroot, but it's actually because bloodroots have roots that are as red as blood. It's a very creative name. And the root right here, or the rhizome, if we want to be fancy, it's usually harvested and it's used to treat fevers and ulcers. Red trillium, however, has a wide variety of medicinal properties depending on what part of the plant you are consuming, but it's mostly used as an antiseptic. The most commonly harvested part of the plant are the leaves. Now, both bloodroot and red trillium are myrmecophorous, which is just another fancy word for saying that their seeds are dispersed by ants. Ants are attracted to the elaiosome right here, which is that fatty appendage of the seed that is shown here. And because both of these flowers are commonly harvested, we wanted to determine its range, and we want to see how it may be impacted under various climate change scenarios. So to figure this out, we had to obtain two different types of data. So the first data that we had to get is called occurrence data. Now this data was obtained from gbif.org, and it's just a database that collects records of where the species was seen. Once I uploaded this data, I had to do a quality check on it because there were thousands and thousands of data points, and we wanted to make sure that all of the data that we were looking at was accurate. So to clean it up, I had to manually remove any points that were out of its range of interest that we were looking for. So in this case, this is for bloodroot. It's focusing on the eastern United States. Mostly bloodroot is seen throughout the whole span of the eastern United States, but we do see that there are a few occurrences out west, if you can see right here. And those occurrences were probably either planted by someone or someone messed up in the record. That might not be the right longitude or latitude. So we had to remove any points like that because it's not its correct range. Same thing goes for red trillium here. They're mostly seen in the northeastern part of the United States. And again, we do see a few occurrences out west for the same reasons as bloodroot. So I also had to remove those points as well. The next stage in the cleaning process was that we had to remove any points taken from botanical gardens, herbariums, or anything that was cultivated, simply because we wanted to see only specimens that were out in the wild. And as I mentioned before, that's why we removed any points that were out west. The next data that we had to get was environmental data. We obtained this data from worldclimate.org, which obtains a set of maps that describes the rainfall and temperature around the world. And I use this database to collect past, present, and predicted future climates. Now, for my research, we had to do something called ecological niche modeling, which is also commonly known as E&M. And that's just where you take your occurrence data and you take your environmental data and you combine it, and then it's going to predict the range of the bloodroot and the red trillium under the various climate change conditions. Now, that may sound like a pretty straightforward process, but it actually is pretty extensive. So once we obtained all the occurrence files and the data, everything that we needed, and we cleaned it up, I had to run it through a software called RStudio. Pictured here is what is called a script. And I had to enter in commands that basically would tell the software to tell us what the expected range of the bloodroot and the trillium would be. Once when we got those predictions, we had to upload them into QGIS, which is shown here, and it maps out the range for those, and it gives us the visual representation that we're looking for. So to kind of start things off, we can start off with bloodroot. So here is a map that shows the current suitable habitat range for bloodroot. The darker green means that there's a higher probability of them being there and that it's a higher suitability for them. Where the map shows areas of lighter green, such as in the Midwest, that just shows that they could be there, but it's not really going to be like a highly suitable area for them. But they could theoretically make it there. That being said, they aren't really necessarily going to be there right now, but like I mentioned before, both bloodroots and trilliums are dispersed through ants. So that's quite the journey for ants to make it from, let's say, Virginia all the way out west, so in the Midwest. So it might no longer be suitable for them by the time the ants actually get there. But if you can see how relatively spaced out the green is through the map. And then in this scenario, we predicted the range of bloodroot under mild climate change conditions by 2070. Now the range has become less suitable in Virginia, if you could see, because it's not as dark green as it was before, and it's actually starting to move more northward. Here in this scenario, we predicted the range of bloodroot by 2070 under extreme climate change conditions, and notice how much the range has shifted from its extreme to its current. So if I can show you right there, it's pretty severe. And it just kind of shows that the suitable range does show that they would be very well off in its current range. But then as the scenarios get more extreme, it's moving more northward and away from Virginia and towards Canada. So then we repeated the same process for red trillium and got the same results as bloodroot. So here is its current range for its suitable habitat under mild conditions by 2070, and then extreme conditions by 2070. So we're seeing pretty much the same trend here where it's becoming more northward and it's not really suitable for an area here in Virginia. So based on our projections, I think it's pretty safe to say that climate change would definitely cause a major impact for the ranges of both bloodroot and red trillium. Now our next big question is, are we seeing a noticeable difference in their ranges today? So surveys have been sent out to those who live in the Southern Appalachia and who are currently harvesting these flowers. Any of the points of the right of the mean, so 50, here's our time of whether or not if they're seeing it, or if there's an early change in the harvest season, if there's no change, or if there's a late change. So everything to the right of 50 means that they're harvesting it later. And then the higher the points mean there's more of an abundance of the bloodroot or less of the abundance. So right here, this is the data for bloodroot. Same question was asked for red trillium. While we may not have any responses, we still see a slight little trend where there is later harvest seasons, as we can see here, because it's of the right of 50, but then the abundance has not really changed that much, maybe has gone a little bit lower than what we're expecting. Obviously there's still many questions yet to be answered in this ongoing study, but for now, all we can confidently say is that climate change does definitely play a role in the ranges of these Appalachian wildflowers. And with that, I would like to say thank you so much for taking the time to listen to my presentation. Thank you to Dr. Jordan Metzgar and the Metzgar Lab for supporting my research and providing any feedback, Caroline for starting this project, Jake for creating the script, and then the Virginia Tech Biological Sciences Stacy Smith Undergraduate Research Excellence Award for funding this research. Thank you so much, and I appreciate it.
Yep, so that was Olivia. So that was actually late breaking results. We just got to that point in the project pretty recently, so we'll be continuing on to do some more things with her data set this spring and summer. But the last talk is about a fern. So my background is in fern biology. So this is always one of my favorite groups, but this is on the distribution of a couple of ferns in Virginia, the maidenhair spleenworts. So this is Sydney Corral's work. It's actually been picked up by another student, Peyton Smith, but unfortunately, she wasn't able to add to the presentation as she got COVID right before spring break. So she's feeling better now, but we're only going to hear from Sydney tonight.
My name is Sydney Corral, and I am a senior wildlife conservation major. I am excited to tell you about my research on developing a range map of Asplenium trichomonas subspecies across Virginia. Asplenium trichomonas, or the maidenhair spleenwort, is a rock-loving fern with a wide geographic range across many temperate regions, including Virginia. This taxon has multiple cytotypes with both diploid and tetraploid subspecies present in North America. A cytotype is a member of a species with different number of chromosomes than another. The diploid cytotype contains 2 pairs of each chromosome per cell, and the tetraploid has 4 pairs, resulting in larger spores produced by tetraploid plants. Diploid and tetraploid individuals are difficult to distinguish and often mistaken for one another. Though they may prefer different habitats, spore size is the most reliable method to distinguish the two. Ferns use spores to reproduce instead of seeds or flowers. The sorus, pictured on the photograph to the left, is a cluster of sporangia located on a fern's leaflets. The sorus is surrounded by a flap of tissue called the indusium that protects the sporangia. The sporangia, pictured in the middle photograph and located inside of the sorus, form and disperse the spores. The spores, pictured in the photograph to the right, are released by the sporangia to reproduce and become new ferns. In order to determine the cytotype of Asplenium trichomonas, I measured the spore size. I used 68 herbarium samples of Asplenium trichomanes from Virginia Tech's Massey Herbarium and Lynchburg Herbarium to construct a range map of the diploid and tetraploid subspecies. For each specimen, I used forceps to remove the sporangia from a leaflet and placed it onto a microscope slide with glycerol. Using a Zeiss Primastar compound microscope, we captured images of 20 spores from each specimen, resulting in 1,400 total photographs. Using image J, each spore was measured across their widest point, including the perispore. The perispore is the outer covering of the spore, shown here as the darker ring around the perimeter. Tetraploid spores are significantly larger than diploids, with an average longest dimension of 41 microns, while diploids are approximately 29 microns long. I combine this information with locations provided through the herbarium specimens to construct a range map. I mapped the locations of each of the 68 specimens by county. Looking at the map of Virginia, the red represents counties with only diploid populations, the blue only triploid, and the yellow with both. The gray color represents counties in which we had no samples. Of the 68 populations, 57 were diploid and 11 were tetraploid. 6 of 7 tetraploid populations existed in areas also home to diploid populations. This map also shows the topography of Virginia, with darker areas being higher in elevation. We were able to determine that while the tetraploid cytotypes seem to prefer the ridge and valley region, the diploid cytotype also exists there and is the most common across Virginia. Out of all of our Virginia populations, most were diploid, ranging from 24 to 36 microns, with an average size of 29. Of the 11 populations that were tetraploid, their measurements ranged from 36 to 46 microns, with an average of around 41. In addition to these findings, I plan to also map the subspecies populations with their elevations in order to determine if there is a correlation there. Through understanding where these subspecies are located, we can learn more about this fern, such as the relationship between ploidy and elevation. Thank you to the Virginia Tech Massey Herbarium for providing equipment, a workspace, and samples. Thank you to the University of Lynchburg Herbarium for loaning me samples as well. Thank you Amber, Chrissy, Crystal, Dias, Jillian, Hannah, Jake, Catherine, and Sarah for all of your help, and thank you Jordan for helping me with literally everything and being an awesome mentor.
Yeah, so that is Sydney's research. So those two subspecies are pretty much impossible to tell apart with the naked eye. So using that spore size is about the only way to tell them apart. So it's hard to sort them out in the field. But that's all we had for presentations for the herbarium. If there's any questions about the student work or anything, I can take those, or we can turn it over to Brenda here again.
Is Mary Rhodes still on?
I am still here.
Oh, do you
I'm
want to?
awake.
Oh, okay. Do you want to talk about your voucher specimens that you've found? Because I put those in one of the newsletters, but maybe people
My
haven't.
voucher specimens for what?
I thought you did voucher specimens at Clear Lake. No?
I don't remember anything about that.
Okay, then.
I mean, is it in the newsletter as a little article?
Yes.
Oh, well, then I guess they'll read about it and so will I.
Well, that was a while ago.
I know, a long time ago. I don't even remember.
One of them was on logs in the lake.
Oh, you're talking about the new flowers?
Yeah.
Yeah, that was Ludwigia. Ludwigia something or other. Yeah, I think I'll have to read about it. It was so long ago.
Okay.
It was really neat. All I want to say is my background was really plant ecology, and what was so interesting was that species had never been found like farther west than central Virginia, and as best as sometimes the seed can come down a river or whatever, but that plant was found on a floating log and doing really well in Claytor Lake. And so we don't know if it's birds or what, because it obviously couldn't have come downstream because the new river starts, down in North Carolina and not in Eastern Virginia. So it's interesting. Yes. Ecologically, how it got there. It could have been on a boat, I guess, and maybe settled on a log. Who knows?
I have a question about ferns, since Jordan is expert. When ferns reproduce, where do they do that in nature? Do they have to do it in mosses? It has to be damp, but where do they it has to be two ferns close together, I guess, so the spores can cross. Where do they reproduce?
It gets there's all kinds of options basically, but so they have that sporophyte generation. So that's the big leafy thing you see out in the woods. And then on the underside of its leaves, it makes little spores. So those are haploid. They have half as many chromosomes. And those little spores, they get launched out into the air. The sporangium in those ferns, it actually has a natural catapult. When it dries out, it'll tear itself apart and then fling the spores into the air. and the spores are tiny, 30 microns long. So some of them might land right next to the plant, but then some of them might catch a wind current and land thousands of miles away. And then when that spore germinates, it becomes this little gametophyte. A big one might be the size of your fingernail, and that gametophyte makes the egg and sperm. And then once there's fertilization, it'll grow a new sporophyte, a new one of those big leafy ferns out in the woods. But then that gametophyte, it's got a few different options. There's a few ferns that can fertilize themselves. Like you would only need a single spore to land and become a single gametophyte. And the sperm and egg from that single gametophyte could fertilize each other. But then for most species, you need two gametophytes from different spores. So if it's nearby, if you have a big population of lady fern and you're looking a few hundred yards away, there's going to be a lot of spores land there. It gets harder when it's way far away, like if it floats out across the ocean and lands on a new island, then it's harder for it to reproduce. But as for where the gametophytes grow, they really like freshly exposed soil. So if there's a new road cut in the woods or a new landslide or something like that, they really like landing and germinating on that fresh soil. Even a year or two later, they won't germinate as well there. But they usually do need some kind of shelter. Like most of the species need a little bit of protection from the sun, and then they need most of them need a little bit of water for fertilization. Does that answer?
So, yeah, and so that's fascinating. Well, one time I went to a talk and it talked about, it was a guy who was a specialist on orchids, and you know how the trees fall in stadium woods, they make those falls, and there's this big hunk of root and earth. He said that's the best place to find, that's where orchids will germinate or something. Something about that soil was different. So is that the kind of soil they might use?
Like right where the tip up mound is, like yeah, especially shortly afterwards. There's kind of a ticking clock on it for ferns, it seems like. So after a couple seasons, the spores probably wouldn't germinate as well there, but for the first year or so, the spores would germinate pretty well on that kind of spot.
Any other questions for Jordan?
No, but it was great. And yeah, I think it's so wonderful we can hear from the students.
Mm-hmm.
It's not easy what they're doing. Yeah, and someday we'll have Jordan tell us all about ferns. It's really tricky, those crazy little ferns.
They're a fun group though.
Okay, well, thank you very much, Jordan. I learned a lot from your talk next, especially this fern thing at the end. So very interesting. So,
Thanks for having us.
mm-hmm. Okay, so our next talk will be in April, and as I mentioned, it'll be from the Blue Stem Nursery, and I'll send you information about that then. Well, everybody enjoy the spring weather as long as it's here before it goes back down to winter and then back up to spring. So, okay then, thank you, everybody.